role of ERBB2 in cancer and cancer treatment. So what we have to do is, it is mentioned like activation of ERBB2 receptor signaling pathway. So the pathway is going to be ERBB2. So this way. So what we will do is, we will open the actually, I think I told you 2 or 3 sites I might have shown you. So suppose you start from the target directly, what you normally do is you will start the open target, and from the open target you will proceed. So suppose your project is something that is connected to starting from this. Otherwise, what you do is, this is a pathway. So you can use Reactome, REACTOME, reactome.org. So this is a very wonderful pathway database. And actually, your query you can give here. This is actually ERBB2 receptor signaling pathways. And cancer, of course, cancer related, that is, so, and cancer. So we have these results here. So you can take ERBB2 receptor associated signaling pathway. Then you can see that this is the signaling by ERBB2. So here also we have to see that the reactome, actually the species ensure that only human species has been taken up. Similarly, normally you will get all these. Otherwise, sometimes you may have to specifically mention pathway, because here that right from the query it will understand signaling by ERBB2. So you may open here. So click on signaling by ERBB2. So we will have this, and here you click signal transduction Homo sapiens. That mechanism is already shown here, but let us click here, signaling by ERBB2 Homo sapiens. Then the pathway related information will be completely obtained, mechanism related information will be obtained. So actually this is the mechanism, something related to that. In your paper you may have to include that. Now you may click on molecules. So we are having 56 proteins. So click on this 56 proteins. Then all the proteins, their identifier, etc. are given to you. You may download it, and in the downloading you don't require that name of chemical compound. What you normally require is you are having only, you need only that protein information. This is a protein information which you are having. So here also you may change, you need only that identifier, other things you don't require. Now these are the identifier, protein identifiers are included here with this query. So you may select them. Then if you want you can save it, Control C. Now what we do is, we will open the cytoscape. And I told you cytoscape is going to be the platform which is used for generating the networks, required networks. Then, and from the information, from the networks, whatever information which you are getting, this will be used for coming into certain conclusions. And so your work will be like that. So once you select the molecules, protein molecules, then you have to generate the protein-protein interaction network, what do you call otherwise as PPI, protein-protein interaction network. And this will be generated from cytoscape. And now cytoscape is open. What you have to do is, you can give the view option as full screen. Now this is the full screen. option. Now click on that apps. So we have the app manager open here. So click on app manager, and whatever apps which are available for Cytoscape, that will be loaded automatically. And these are the various apps. 238 apps are there in Cytoscape. Then, so what you require for this work is basically string is required, stitch is required. So these are some of them. Then you may require CytoHubba, and CytoHubba will give you details of topological characteristics, topological and some of the I mean molecular characteristics will be generated. So the major requirement for you would be the basic fundamental thing is string. You have to see that string is already there. So let us see whether string is there. The string app, here you see string app, it is already installed in my system. But when you work it, then see what you see. I will show you. Suppose I need to install strongest path. What I need is, let me click here. Then I think you might have seen this view on App Store, then install. These are the two options which are open up at present. And now what you do is, you may click on install. So automatically that could be installed in this manner. So this is the procedure which we follow normally. Okay, so that is what I want to add here. So whenever you need to install something, this procedure will be followed. Then what I told you is I need string that is already installed. Then I may require CytoHubba, and that is also installed in my system already. CytoHubba, that is it is not at install. So let us install CytoHubba, mind you, CytoHubba. So let us install it. Then okay, now we have to see whether the Wi-Fi layout algorithm, whether it is installed or not. So that is also installed in my system. It is already installed. So these are the minimum requirement for us. So you need string, then you may require CytoHubba, then you may require Wi-Fi layout algorithm. So these three apps you install initially. Thereafter, so what we do is, we will already you can see that string is there. So either you can take string, or what you can do is you can open from here directly network. Import network from public database. So you can click here. Thereafter, so what you do is, here if you open, you can see that stitch protein or compound. Protein query also you can use. So this is another method for putting your data into this. Stitch, as for example, if you take stitch, then here you can give the protein. Already I have collected the proteins from a reatome. Now that confidence level also, if you want, you can change. By default, it will be 0.4, because biologically a probability or a confidence score above 0.3 is feasible. So actually speaking, biological probability is 0.3. If it is going to be 0.3, we say that it is probable. Mathematically, probability is actually 0.5. Anything more than 0.5, we say that it is probable. Less than 0.5, we say that it's not probable. But biologically speaking, probability also comes in that level. So that is why by default 0.4 will be there. Now another thing is, suppose it is going to be more than 0.7, that is that definite. So whatever result which you are getting, that will be highly reliable. And let us see that, okay, 0.7 is going to be put up as a confidence level, because what I need is the data that is put up here, that should be highly dependable, and we should have a very high confidence level for it. Now you may click import. So the network will be generated, and this is the first network which you are seeing, and generation of network which you are seeing with the 46 identifiers. Yes, please. Okay, okay. Let me check. I think the is it visible in between? What is it? What you are seeing at present? Yeah, network you are seeing, right? Okay. Now, if I mean, I mean, illustrating it. So if anything is stuck in between, you have to tell me immediately. Okay. So this is actually the network which you are getting for ERBB2 related network. Okay. Now, suppose I put the, I mean, now you can see that at the bottom there's a table. This is a table actually. And before going through the table, what you may do is you may click on tools, then analyze the network. Let us keep it. So analyze the network. So you'll be able, you can see that one I mean basic analysis screen is put up. And if you click here, you can see that it is a float option. So this result will be floated. Now what I can do is I can keep that result somewhere else if I need. I can make it in this manner. So these are the results which I am getting. Number of nodes, number of edges, then average number of neighbors, network diameter, network radius, characteristic path length, etc. And this is a basic result which I am getting. And so that you can note down immediately. That result you can save or you can note down immediately. Then I mean, then go through the result basically. And I think you don't have to make any of these in the beginning, but we will do it one by one. We will do it later. And once your project is over, you will see. Now let us go through the result which is given in the table. You see that results, so many values are there, and here among these results, one of the important results is what you call as degree between us, etc. They are going to be very very important to us. And right from the, so after some biological results, so these are the I mean molecular properties are generated. In the beginning, molecular properties are there. Now starting from I mean the yeah, eccentricity always, you are having the topological properties, and you see that this is a topological degree, and betweenness, etc. These are all going to be very important to us. And so what you need is actually you have to import table from file by clicking here. So it will be saved on a I mean, I think we can save it for the, yes please. I don't know why it is blurred, because the my screen is shared fully. My screen is shared and it's actually going on, and I'm working out now. So if it is blurred, I do not know. It may be some relay problem or something like that. Because even in the video, it is not blurred. If you can just adjust, then video I will share today itself. If possible, I will share the video. Okay, is it all right? Because I'm saving it because this is a project illustration to you, and so automatically, if any such difficulties there, I think I will share it as early as possible. Then, and when I go on with the project, you will also get some idea about yourself sometimes you'll be able to do it. So desktop, then in the desktop somewhere we can save. So let me create a new table, then BME projects, BME project. Then what I do is I save the results. I mean, I think that name is gone now, desktop. Let me check whether BME project, yeah, this is the BME project. Then here actually ERBB2, ERBB2 results, ERBB2 topological, and so let me write cytoscape, cytoscape stitch. I used stitch. Then no, here actually that what I need is control A. Here I open the file. So once again let me check here file name. Now what did I click here? Import. I clicked import instead of export. Now what we do is BME project. Here you may put the, I mean sorry, control research. So what I do is ERBB2, ERBB2 stitch result in CSV format. So save it. So that is saved. Now, so what you do is, so you have got the primary database, then you can go through that result. So control D, I mean shift D. Now let me take the BME projects. This is the result which I have got. And this is the total. Then here that, I mean, identifier is there, then average shortest path. So like that the entire result is recorded here. Now you can go through the results, then degree wise you can find which one is most efficient as far as degrees. See, as degree in fact is actually the number of, I mean, edges to which that is connected. It's actually a measure of its connectivity. So into that, as the degree increases, its interaction possibility increases. So that is why we say that, so that protein molecule with maximum degree, so that will be a very suitable compound for us. I think up to this, you can, initially you can complete in your project. Then let us take the second project and show how that project could be done. So what I do is I will pick the second project. So only a small work I completed so far. What I did is ERBB2 pathway is generated. Then from the ERBB2 pathway, related protein molecules have been identified. Then the already you studied top gene, so automatically you can give same data to top gene. Then you can do the gene I mean annotation, then pathways etc. connected to these protein molecules. That also you can do side by side. Now in the meantime, let us see the second problem. Second problem is bringing the importance of WNT signaling in cancer. So what we do is WNT signaling in cancer. This is going to be our problem. So again, what I do is I will open the reatome. So this is the reatome phase. So what I do is in the reatome, this is second problem I am taking. Control V, Control V. No, it is WNT pathway and WNT pathway and cancer. let us see how many protein molecules are there. W and D pathway regulating and cancer. So this the first one is the W and D pathway in cancer. W and D pathway related cancer. And this is a pathway, and click on the disease Homo sapien, signaling by W and D pathway cancer, and the corresponding pathway will be generated in reactome. And here also, mind you, suppose you want to check the expression. So let me add one more point here. What I want is, I want to see how much WNT related protein, how much that WNT will be expressed in the gene. So already you did gene expression profile, so that result will be generated here. So WNT related gene expression profiling, that will be generated here. So we'll be able to see how much this is generated. So this is actually the same thing which you did there. So these are actually the corresponding protein molecules which are obtained. So the same result you are getting, whatever you did on the expression atlas, same thing you are getting. So you'll be able to see that among them, among the selected protein molecules, or among the protein molecules included here, which are going to be highly expressed. So that idea also you will get, which you can add in your paper. When the expression profile is analyzed, you will write it in that manner. WNT pathway, similar to ERBB2 also, you can do it in this manner. So what you do is, the WNT pathway related protein molecules have been identified with the help of reactome. Then while analyzing the gene expression profile of these, it is found that these are highly expressed. And if required, what you can do is, you can download that expression profile, and from that you can draw the box plots, and you'll be able to say that, okay, these are the protein molecules which are expressed. Because your paper means you have to make it as informative as possible. It is not just working out a problem and getting answer from it. So when you do when you do a paper, what you have to do is, you have to make it with a required depth as well as width. Width is also important. And if you just focus on a particular point, actually that alone may not be sufficient in most cases. You will make the proper width. So that is why I told you, expression you can put, structures will be there. What I want to see at present is the number of protein molecules, connected protein molecules. What I see is 34 protein molecules are there connected to WNT. So let me click on 34 protein molecules for doing the network analysis. So these are the protein molecules with their identifier. Now let me download it. Then what I need is, I need only the protein. Other things should not be there. So let me take the protein alone here. Thereafter, the my required result is only the identifier. So you can save it if you want. You can save it, because in your paper you can give it as a table. But let me click Control C. Then again I will go to Cytoscape. Let me open Cytoscape. So this is the first work. Now what I do is, let me use string instead of stitch for the time being. So this is string, means what I need is, let me click here. So next to string, if I click, one window will be open. Here let me add Control V. Then so what I need is, I click here. That is actually the search network. Then click here. So we'll be getting the 34 identifiers are there. We'll be getting the network with the 34 identifiers. So this is actually the network which you are getting. And so what you see is, actually some of them are highly connected, some others are not that much connected. And I told you how to make changes on that. You have to click on style. Then so you will be able to change the label. You can change the shape. You can change whatever you want to change and make it into a publishable form. Then you can save it. So that part already I told you. And so this you can do, but mind you, this is highly optimized, so no question of you keeping few of them. So here you have seen that this protein is least connected because it's just outside, only two connections are there. This is that protein basically. And of course its degree etc will be comparatively very weak. And we have seen that the highly connected protein molecules have to be generated. So here also what I do is I will click on network again. So the parent place, parent thing is already there. Thereafter tools, click on tools, analyze network, then okay. So I get the result in this manner. Now what I do is I can get the result, I can read the results. Now I can keep it anywhere or I can make it full screen. Then these values can be recorded.
Problem which I am going to take up is
what? Yes.
Using PPI as well as PDI. PDI okay. I will take after that. So let us see these four cancer types.
Is it working?
Breast cancer, ovarian cancer, then I think lung cancer,
I think you
and colorectal cancer. So these are the major four types of cancer.
So this is actually the network.
So what we do is we will identify
which you are getting.
Common.
Now the first thing is I told you
among
three types
cancer,
of results you have to generate. The molecular results,
then lung cancer and ovarian cancer,
topological results, functional results, etc.
So we will do
to be
PPI
released here. So this is the result which you
protein interaction.
And if you just go through, you can see that all the results are there.
Then if required, again, we will do the co-expression and physical interaction studies.
Export it.
We will
so you
generate
here, export.
the topological properties
I think
using cytoscape. So that will be our
SBME project. Same page.
So similar to that,
it is going to be
similar to breast cancer. Breast cancer itself, that is of different types. That you can take. Similar to
first thing
these four examples, you can
take. In the meantime, suppose I want to do the
cancer and
top
see
gene
how they are going
so
to
what
be
I do is I will
then
open the top gene.
So that what can be extended in that manner. But to start with, what I do is I will take these four types of cancer.
Top gene
then
I can
see whether there is something common. And so what we believe is actually cancer is a syndrome which would be produced or which would be there in the body before 10 to 15 years. The disease
so
is
your result, say you need to concentrate only on one problem.
Few mutations
so those about 20 problems
which organ is going to be affected.
Then click on top gene.
ERBB2, then TP53,
so what I do
they are all
is
common
I will
mutations
add same
found
results
for
here.
all types of cancer.
Then this is something
sometimes addition of
testing set is there. If you want, you can put few of them there, or you can
something
put
else,
some other protein which are connected to cancer like BRCA1,
so once a syndrome is there,
BRCA1,
once a tendency is there in a human being, then depending upon that most liable
BRCA2,
or most what do you call as susceptible organ,
yes,
that organ will be affected. As for example, if the colorectal region or if the
is it audible now?
Head and neck or what you call the alimentary region, if that is it audible? The most susceptible region, which may be sometimes resulted out of the habits, environment, food habits, environmental habits, etc.
Got it.
A person who is more susceptible to lung cancer. So a person
what I told you is after this you may go to the
then
so top gene
or a person who is
the top gene also what you do is you give this
so such a person will be highly prone to lung cancer
in the training set. And
then ovarian cancer, that also.
Then training
cancer
patients, if you want, you can give few
may
of
develop
them.
ovarian cancer
and
few
of them can be given.
So they are actually that environmental factors
the
or
problem
sometimes our lifestyle
with
that may
BRCA1
decide
instead of
which
BRCA1
organ
I put
will be
something else.
affected at the
Then
end.
so what you may
At the
do is
same
you may
time,
go back,
all these cancer types will have some
then
common
give some
mutations.
of
So
these
what we
queries.
would like to see in this
Few of them
illustration
you can
is
give.
to check the
BRCA1
common
instead of
mutations which are there.
What I put
in
is
it.
BRCA
So I
exclamatory
will
mark.
use some
Then
open target
thereafter,
as for
let us
example
submit.
to generate the respective genes. So what I do is
Now the problem is actually
I
this
will
identifier is not
with,
accepted here. So automatically you have to change them into the
open target
because what you need here is in top gene, what you need is you need this identifier. That means actually that name has to be given. Entries,
Open target
otherwise
form.
entries you have to give. These are the two names
Then-
which are given to.
So we have actually
So what
types
you
of
suppose you
mutations.
want to do
So
it
let us
again,
take,
then what you may do is you
find the
may
target
change
for a given
you may identify name from NCBI. That
disease.
protein name you have to take.
So
then
first thing
only
is,
you will be able to
so
do
let
it
us
in
make
top
lung
gene.
cancer.
So top gene, similar to that
Lung
top
cancer.
net also, same manner, same problem will be there in top net also.
7,367 targets are
So let me
associated
open
with it.
the network. Now here I use the
If you
first
use
method,
open
which
target
I use
platform,
stitch, not string.
then
So let me try stitch and string. Both
here
are
also what I
equally
can do
possible.
is I can make
So what I
the
do
confidence
is import
level
network
1,
from public
and
space.
with the
Then
one. So
stitch
first
is
thing
already
is
there.
let me
Here also I can
download
have
the
stitch
entire
or string. Then stitch, give the query. So sorry,
mutations
the
or
full
genes
name is
responsible
not
for
included
colorectal.
here.
Yes,
So full name
lung
I have to
cancer.
take.
So let me
So
put it
from
lung
DME
cancer.
project, this is the
Then
second.
the next one is,
one.
so instead of lung cancer, let me put breast cancer, which already we have. I don't know whether we
Here the identifier is already there.
saved that cancer.
This is the identifier. Second one,
Breast cancer,
ENS
9,547 or something.
So the result, I think you
So
can,
many
you can directly
mutations
go to
are there.
when the top gene
9,157 are there. So that also you may download.
top gene. You see the required input is you have got two options. Top gene. You can give no entries a number you have to give. Okay, so that is not possible directly
Then
from
the
here.
next
But
one is,
here you are getting the
so lung cancer
identifier
we
in
have
this
done,
manner.
breast cancer we have done,
But
ovarian cancer.
Earlier I included
Ovarian
few of them
cancer.
have been included. About 34 molecules are generated
5,371 genes are there
entries.
associated with it.
Yes, these are the molecules which I didn't save anywhere.
That also let us save.
When you are to save the result, what you need is you can download the result and you can save it. Now this result I am giving to Cytoscape, and in Cytoscape,
Last one
what
is
I need
colorectal
is
cancer.
I need a network to be set up, and my confidence level is 0.7, which I reset. Then
Colorectal cancer.
if I import, I'll be able to get the network. There's a third questions network. Network is already obtained.
So actually
This is the
got
network
in
which I am
3
getting.
different,
And
4 different
you can make
Excel
the different changes
as
by
CSV
clicking on
format.
the style, fill color, and make it. You can label if you want to relabel it. Then transparency, etc. You can change if you want. Then you can save it. So when you save it, you have to click here.
Breast
Export
cancer
to
already.
files. Okay.
Breast cancer.
So you may click here.
One
Export
is breast
as
carcinoma.
network. Export as image.
We already saved.
So you can put it as export
Then
as image.
let us
Then
take the first one.
that as
Target
PNG
associated
you
breast
can
cancer.
save. PostScript also you may save, because with the PostScript you will be able to make further modification with the PostScript. Then PDF also, if you want, you can save it as PDF also.
So what I do is
you can save it. You can zoom the image if you want. You can zoom the image. Pixel also you can change. You can make it as high as possible. Then you may save the result also. So here also
these are the results.
tool, you click on tool, click on network, okay,
press
you
cancel.
are getting the result. If you want to read it separately, what you do is you may click here, then it will become float. Now you can keep it anywhere, and if you want it,
Here about 700 are there.
you can make it
623 up to 1.
So you can read these values,
So let me save
note
it
down these values. Then
another
thereafter again you may go to
control
the network.
So these are the
Then
common.
see that the results are well displayed
With a
well
high
included.
confidence
So
level.
these are the results which you are
So
getting.
they have been saved.
You are getting the entire results. Then export the results.
Okay, 621
You may
or
export
something
it.
like that. Then let me save it. Save.
So here what you can do is actually stitch network. Then stitch network actually here also we did stitch.
Next
Then
point is colorectal cancer.
Problem is what you have done is actually you have taken the third problem, right?
Cancer.
Third problem is what was the gene involved in it. So gene wise you have to save it. Then
this is colorectal cancer.
So you WNT, not third one, WNT.
First one is actually target gene.
Yes.
That title is given.
You may save it in that manner with the WNT.
Here also,
before putting it in this manner, CSV, you may put it. Underscore WNT.
There. Control
C. Dot
that
CSV.
Also, let me save separately.
CSV file.
And this is going to be
result.
Major data.
Then you may identify the protein molecules with high degrees. Then so these protein molecules will be identified as most suitable for your
taken in that
further analysis. Now the third problem is actually regulation of gene expression by
starting from EGFR,
hypoxia inducible factor. Hypoxia inducible factor. So what you need is, this is hypoxia related. So you may go to the reatome,
Control
the
C.
same manner.
So let me save it here.
Let us go to the reatome database.
Control V. Save.
Here I put the name hypoxia.
Last one is ovarian cancer.
Inducible. Hypoxia inducible. Regulation of gene expression by hypoxia inducible factor. This is the example which I included here. So you may click on that, and the corresponding mechanism or pathway is fully shown here. And that is not fully visible. At the same time, you can save that
POLA to up to that.
Then you can put it as PDF also. And from PDF, if you want, you can change PNG. You can save it as PNG with a high quality, etc. So you may save it in the required format. Then with that,
now if I want to check the common genes
with the help of Photoshop,
two techniques are there. First
so
technique is
possibility. So click on
I can use
what I am telling is
the
regulation of gene expression by hypoxia inducible
Venn diagram.
Click here.
That is here available.
Here also you will be able to generate all these molecules.
Venn and Euler diagrams,
structures,
and let me install it.
Then analyze. Thereafter, you can download. You will be able to analyze that result also directly. Then, so let us see the expression here as far as hypoxia related expression.
Let it restart.
So you will be generating the expression atlas. You will be getting the expression atlas with the expression on this particular gene. Then other values also. I mean, all the molecules are included here. So this, if you download, immediately you may do the box plot analysis.
I can
and based upon the box plot, you can say which all proteins are well expressed. Then
so what we need is actually we need
go
to
the structure. Let us click on the structure. Object does not contain associated structures. So no additional structure or associated structure is there. Now molecules, if you click on molecules, you will see the corresponding molecules. 11 protein molecules are there.
So we have.
Then analyze, if you click on analyze, analysis results are shown here when an analysis has been run. So you have not done any analysis at present. At the same time, here actually on the top you might have seen, here you can have the analysis. So here is the option for your analysis. So you can click which analysis has to be carried out.
Control C.
If you want that data to choose that data and do some analysis, so you can complete this work. Then you will be able to analyze that
Here.
data directly,
Control
and
V.
You can download the results
This is
as
breast
shown
cancer. So BC.
earlier.
Let me put it
So
as
here
BC.
also you will get a description of that,
Then
what it is.
Second one.
Then some idea about, along with the major references.
Let us take
That information you will be able to get.
colorectal cancer,
And
CRC.
finally, if the molecules are put up here, you are having 11 protein molecules directly connected to it. Now, once a protein is identified, let us immediately make the protein interaction network, PPI. And here also I generate only identifier. So I'll be getting these protein molecules generated. Control C.
So CRC.
Control C. So once a protein is chosen in this manner, then you may go to cytoscape. Again, open either stitch you open or string you open, because as far as network generation is concerned, same thing. Whether you are using stitch or string. Then
lung cancer, LC.
So here, let us do string. Then same manner stitch you can. So stitch means again public database you have to generate string. Then give the data 0.7, same confidence you are sticking on import. Then with the 11 identifiers, you get the molecular network. So this is the molecular network which you are getting.
Another list. So
and here you see that its actual name is there, not the identifier name,
cancer.
HIF3A. And these are all its actual names: HIF1A, HIGD1A, EPAS1. So these are all its actual name. And that display name is shown here. The result, it will be shown with the identifier name. That also will be there. ENSP number is also there.
This is ovarian cancer.
As I told you earlier, you may export the results. So here it is going to be hypoxia related. So what I do is I will put the name hypoxia.
Now what you will do is submit
related.csv. So that result I obtained. Then so the basic thing is, again you have to analyze.
So this is the Venn diagram that is obtained.
Protein molecules which are having substantially high
CRC, BC, IC, OC, 344
degree, betweenness, etc. Molecular properties, etc. Whatever properties are.
So these are the
going to be relevant. These properties you have to put up
which are
in the form of an Excel sheet. Then
shared among
you may have to include these results.
Cancer, breast cancer,
proper
lung cancer.
So what I want to tell you is actually whatever project which you are
colorectal cancer, breast cancer, lung cancer,
almost
125.
But
then
once
colorectal
you
cancer, breast
move
cancer,
on,
ovarian
you
cancer,
see that
5.
your
so like that,
result
different things
is
are
also
there
going to
given
be
to you.
changed.
So your first
Then
trial will be this one.
Next work is actually,
And,
next problem is coordinating regulation of oxygen dependent proline.
Let us take only 10 or
hydroxylation
15 for the time being,
hypoxia
because I
inducible
want to
factor
show you that
alpha.
actually
here
you have to
also
take
what
the
you may do is
entire
you may,
menu. So just to
click
show you how it
you
is
may,
done. So you can practice
take the
yourself.
relevant tails
So
and
cytoscape
give
also
to Reatome.
So let me
Because
start
Reatome
cytoscape.
is a text query based database.
So taking 344 is not time consuming, but
exhaustive
I
database,
just want you to
secondary database,
work it out
which makes use of all primary.
So what you do is
database,
actually you
all
may
primary database. So here you may
find
give
the common,
query oxygen dependent
thereafter
factor
let
alpha
us
1. Okay. So this is the
save them.
required pathway. Then
here also you see that that pathway is there.
Here
And
what
once
I
a
do
pathway
is-
is obtained, you need that pathway to be included in your thesis. So what you do is you will
be
save it
emptied
as
always. It will be there in the
PNG
server only.
or PDF. Then you can, if you want, right from PDF or PNG, you can modify it. Modification means you can
What I want here is
the
string. I want
quality of that image can be further improved. Then you can include it.
string up.
Then what else we need? We need to find the protein molecules, corresponding protein molecules. So oxygen dependent proline hydroxylation. So that factor is there. Now here also you can do all these things.
String
You
is
can
there.
give the expression.
So here let me
Then
add
corresponding protein molecules expression analysis
actually,
will be there.
string
So you
needs them
you
in
can
this.
put the analysis part by expression. Or if you want to do the real analysis, you can
put them in this
click
manner.
on that analysis. Then
Sometimes it
give
may
the
ask you to
analysis
put it in
part to
that
be
manner.
completed with the data. Thereafter, so here also you got the
Which
result.
if you want you can
And
modify it in the
so that already
gap
that is
format
familiar
to
to you.
column
You will,
format using Excel.
once a result is obtained in this manner,
You
the
don't have to
expression
do it
values
manually.
are shown here. You may download
When
the
a
result.
large number is
Then
there,
from
you
the
don't
result,
have to do
what
it
you may do
manually.
is, so you can here all data table content. You will be able to save it as table data. Then from that,
So we have taken almost 10 or 15 only.
box plot.
So
From the box plot,
we can do it
you
manually.
can immediately you can see that the which one is expressed.
So these are actually the common
that
genes
details can
which
be
are
given.
there in
Then
these
molecules,
four
if you
major
click,
types of
let us see
cancer.
how many molecules are there. You are having 66 molecules connected to it. So you may click on the protein molecules. Then while downloading, so what you do is you may download, I mean not that side, molecules. You are getting
take only
these
few
molecules here. Now you may download the protein molecule. Only
you are
protein you need. So let us make a screening in this manner, protein alone.
Now what
type
I
you
want
are
is let me make them
identifier.
into
66
networks.
molecules are there mainly. So with the 66 molecules you are getting the
44 identifiers
network
have been taken up
next time
out of 344 or
using
something.
string.
So this is actually the
here also import
graph that I got.
Network from public database.
Then
Stitch.
here also what I find
add
is
the
few of them are
result.
removed.
Make sure that the confidence
directly
level
from
is
it.
0.7.
Now
Then
we know that the topological
import.
properties of
Within few seconds you will
this
get the
network that will help us in
ready.
separating them. So what I do is actually in tools,
Then
let me do
with the 66
analyze
this is
network.
going to be very clouded.
Means a primary analysis.
So
will be
whatever
carried
maybe
out.
that we don't have
So
to
you
make
are
any
getting these
variation.
results.
First
So
thing is you will do the
make it float.
You will analyze
so
the
that I
net.
can keep it somewhere and read it.
Then the result is obtained as I told you earlier.
So what I see is number of nodes
make
44, number of edges 103, average
bigger,
number of
then
neighbors
read the values
5.
and show the values.
Then
Then
like that primary
the
data
results
I'm
you may
getting
export.
here in this manner.
Then same manner you may export
this
it.
is the
So this is
query
the
which
strategy
I
which
got.
we are following for all these projects.
Thereafter,
I think
what I can do
you
is
have got
if I
some
want,
idea
I can
about how to
get
proceed.
more details.
Then tonight you may go through the entire
by using
project
Cytohaba.
titles. Then I mean, identify one project of interest to you. Mind you, whatever I showed you, that is not the only work to be completed in your project. You have to add so many things into it till it becomes highly informative.
This is actually
now you will carry forward this problem till
tool
your
or
work is
app
over.
which will give you
And in my classroom
the
discussions,
node
I will choose one
subnet information,
which is
node
not chosen by
information.
any one of you.
Then
That is why I've included about 24.
so that
And few of
further
them will
calculation will be there.
Vacant. And one of them I will take, and with that I will show you how to proceed further. Then so you will be able to do along with me, you will be able to complete your work. Then so I think that will work out fine without much stress from your
side, you will be able to complete your project beautifully. I think that is working fine. That will work fine. What do you say? What do you think? What do you feel? My dear students, what is your suggestion? You tell me. Hello? Yes. What do you say? Kirtana? Kirtana Krishna? No, but what you have to do is you may choose a problem. You don't have to take the entire problems. Each one will be taking one problem. With that problem, you will do the entire networking related experiments and come to various conclusions. And these conclusions you will document in a proper manner. Thereafter, we will send it for publication. Okay. Is the table format. So that is why I told you you have to view full screen. Otherwise, sometimes that which are there at the bottom you may not be able to see. Now, these results are the major results which you are getting, which you are using actually. We can use betweenness. These are the topological, basically. Topological as well as some functional results are there. So mind you, degree, this is going to be a major result which you are getting. Betweenness, neighborhood, etc. Everything you got at present. So what you may do is you may export to file, or I mean, I think you can, but you can export table to file. This table you can file, then you can make it as .csv, and let us keep it here the same desktop. I put like a class on the class 7th December. So here, so what I did is the genes common genes network .csv format. Okay. Now, now what I can do is actually it is saved as CSV format. Now we can take that in an Excel, open with Excel or WPS. So whatever you what Vishwaya. Now, all right, right. So this is a result, and here that identity is given, gene ID is given, then average strength betweenness, closeness. So what you can do is the first comparison will be based upon degree. Then you can see that those with a high degree like 18, 13, etc., they are going to be very significant. It's the first information which you are getting. As I told you, for cancer, the major genes will be highly connected, and it won't be at the periphery. It will be deep inside the network. So automatically that will be having a high degree. So you can choose the genes which are having high degree, like 6, 12, etc. They are going to be at the same time. There is one with a 0 degree, so that is less significant. And highly significant genes can be put up in this manner. Hello. Okay, then so this is the first method, first technique to find the major genes which are associated with it. So based upon the degree, you will be able to check. Because I have taken only 44, so only one or two are there with 0 degree, and that also, suppose more protein molecules are added, then definitely I'll be able to get. Then betweenness also, that will also give you some information on it. Then so this is a technique to obtain primarily the PPI, generating PPI. Thereafter, getting the basic or what you call as the functional importance in it. Then, if you want, what you can do is you can even you can take the I mean gene mania and see whether they are really, because we actually we have taken one tool in between, that a common thing. Then so what you may do is you can take gene mania local search, but mind you, gene mania also same problem will be there. So if that gene is added, then only you will be able to check it for gene mania. Gene mania problem is actually it has got all the data sets added, so you can have human Homo sapiens is already there. I added it already there. So when you do it, major problem would be each data set means each organism's data set that will be taking much space for you. So what normally people will do is they will use online gene mania, And so for that purpose, actually, Gene Mania is a function of your favorite gene and the gene says. So what I suggest is actually before you confirm the genes, what you can do is you can once again you can check whether that has got this required gene, I mean gene ontology, then using top gene if you prefer, then you can do that. For each example, sometimes the topological characters or descriptors that also changes. But in general, what we can see is betweenness is a very significant factor. Degree is going to be a significant factor. And I just want you to see Gene Mania. Gene Mania is already there in my system. Already I added for human being, Homo sapiens. But when you do it, it will take almost, 20 hours to completely install, human data set. So it will install that data set fully. Results will be identical whether you use your Cytoscape app or here. Here it will be online. It is making use of human gene interaction data sets, full human gene interaction data set. So automatically the data set itself will be very big, very huge, and so in the meantime, what I can do is I can let me Control C, let me save them. Then so we can generate a few of them from network, from file, import, network from public database. You click here, then here actually instead of string, stitch, etc., what I use is universal interaction data set. Then here let me add the molecules which I have given there. Now actually you are getting different data sets here. Different data sets I can say. IMX, it's actually a protein simulation. So whatever which I need, I can just click here. As far as the percent data is concerned, then this is going to be a wonderful setup actually, because you will be getting most of the results in a single screen, and most of the results in a single screen. Then what I do is let me import, because the different tools which normally gives you results in different screens, but if you use this universal interaction database client, all interaction networks connected to your data that will be generated, and from that also your required data like protein interaction data set or whatever data which you have given, that could be put up there. Yes, the gene mania result is already over. So here, gene mania, this is actually, you can see that these are the major results which you are getting. Co-expression data results you are getting, co-expression, then physical interaction, pathway-wise, co-localization, then genetic interaction, shared protein domains, etc. as far as the data is concerned. Then this is also going to be a very informative gene mania, you call it as. So you can see which all things are co-expressed in the co-expression data set. Actually, that genes are one and the same. At the same time, the edges they have been given. They are actually used in different colors, and so based upon that color, because I have put everything on. Now, whatever is required for us, that alone, if you want, you can generate. So gene mania, that is giving results in this manner. The meantime, I think the most powerful tool which I told you here, that also will generate. So in the during that time, let us take some drug molecules and see a protein drug interaction data set. So what I need is, already I have got the few common genes already I have put up. So let me put it here. Control V. Okay, these are the genes which I have put up here. Now, in the meantime, what I do is from the drug bank, from the drug bank, few drugs which are used for this. So let me put as say cancer drugs. Let me put, or separately I can, what I can do is separately for breast cancer, then ovarian cancer. So that is actually better. BREAST, breast cancer. So breast cancer related drugs, few drugs let us take. Then here itself only approved drugs. Apply filter, then filter by market available. No, few of them let us put there. Let it pass sphere, Control C. So what I do is here, let me put that drug name, Control sorry, no research. lady, let it pass here. One drug I have added. Then second drug is mineral. Okay. Now what I do is, so let me take lung cancer. So you need to take the entire drugs available for them. My idea is which all drugs will be suitable for all of them. Cetuximab, cetuximab, NINDANAB, osimertinib, OSI, MER, TI, NIB. Okay. Now again, let me check colorectal cancer. Colorectal cancer drugs, colorectal cancer approach. Cetuximab, then next one, at least two at least let us take. Rigor Rafinib. Ovarian cancer. Ovarian cancer approach. Niraparib. CLOMIFENE. And so few drugs and few protein molecules are put up. I want to see the protein drug interaction. So it's still going on. So all the results, that is immaterial, because same result you will get from already whatever result which you got out of the reactome, same result again you are going to get here. Then mint, so many other tools also. So 14 going on. So whatever is required for us, that will be sufficient normally. Here, mind you, I have taken only 44 genes. So with that, it is taking this much time. But the advantage is actually every data will be generated together. So normally what you require is actually, you may require, now even chart if you want, you can see the hub proteins, because especially for cancer, you know that the hub proteins are the major contributing. So what you may do is you can use either bingo or chat to generate the hub proteins, and you'll be able to see that these protein molecules, relevant protein molecules which are having high degrees, they will form hubs. So that is actually further evaluation. I told you earlier, those which are having high degrees, they will be normally highly connected. High degree means highly connected. So automatically they can be the hub proteins basically. And you will see that using bingo or chat, if you generate hub proteins, you will see that these protein molecules, they invariably they form these hub proteins, that you can see. If this is taking much time, what you can do is you can, have one more cytoscape. You can start one more cytoscape and there you can put this data. So multiple screens you will be able to generate. Because this platform which we are having is only a surface software. This is going to be downloaded, or this is going to be connected to the server. Through the server or container, the entire service is made available. So automatically, it is just like your two software or three software. If one is taking much time, you can leave it, you can start, you don't have to stop it. But the problem is, again, this has to be initiated from the beginning. So by that time, if the other one is completed, then you can use it. Now, still it is going on, 14. That work is going on. Okay, this is almost over. So that I'll be able to show you that protein drug interaction also today. So when you, confirm the targets, you can use the top gene, then see the functional involvement of each one. Then you can use the geo tools which are there in cytoscape. With that, if you want, you can do it. But as I told you earlier, try to avoid the unnecessarily adding tools to cytoscape, because that will make your system very slow, and much memory will be used by each tool for functioning. So that is why gene mania, if you are happy, okay, let us see the gene mania results directly from your cytoscape. With a single tool you will be able to generate the entire thing. Otherwise, what you may do is, you may, such things you can take from online. I think already that is, yes. I have started another option. Now let us see whether string is there or not. It is just like a new tool which I added, but only thing is, let me use app manager, then my required things normally will be added. Yes. So let us see whether string is installed here or not. String up. Yes. Now string is there. So what I do is, let me open the import, then table from public database. I mean, not table actually. Import public databases. Now let me use stitch. So instead of string, I use stitch. Stitch. Homo sapien. Now give this query. Let me check whether that is. Yes. Here, these are the proteins. At the end, I gave some drug molecules. So my data set is actually a heterogeneous data set where protein is there, then ligand is also there. View F11. Okay. Now what I do is, let me import. That means import in the data set, import in the network, connecting these. This is actually PDI, drug protein database. Here also, if I want, I can make the confidence level like a 0.4. Instead of that, let me put it as 0.7. Then here what it says is actually this MPL receptor for thromboprotein may represent a regulatory molecule specific for TFOR. Whether I need it or not, let me avoid, because already it identifies some mismatch. Then import. 77 identifiers are there. Yeah, it's a heterogeneous network. It is almost done. But the problem is few of the drugs only they are connected fully here, because you know that this is a drug that is connected. Regorafenib. So similar to that, nintedanib is also connected. Few of them are unsuitable for these protein molecules, which are there at the bottom. This is not connected, and this is also, these two are connected. These drugs are connected, but it's not connected. So from the small data set which I use, what I find is the drugs which are used. Now here, actually these are all common basically. So whatever genes which I have taken, they are common genes which are there for these 4 types of cancer: breast cancer, colorectal, lung cancer and ovarian cancer. So for which these 2 drugs are found to be suitable, especially this drug regorafenib for the upregulation of KDR, then we can say NTRK1, then again FGFR1. Now, if you want to check the mathematics, or I mean the statistical, mathematical as well as topological factors, what you may do is you just give the analysis network, so you will get some idea about the tool. So how many nodes are there, etc. You are getting that information. Similarly, you may click on CytoHubba. Now that result is automatically obtained. Now what you may do is you may click on this data. This is the data which I am getting here in the table format. Now I see that all these are also having the same results, like whatever results which I got between us. And so through degree, that is a primary classification. So I'll be able to see that few of them are having very high, very heavy degree. So even for the drug, I'll be able to check whether this is suitable. Say two or three things I can generate. One is these are actually the drugs which are suitable for these 4 types of cancer. Moreover, each drug is suitable if and only if, or it is suitable for a particular gene upregulation. So when I use here the drug molecule, because these are connected basically. So this has got a degree of 3. So automatically this is connected to 3 different protein molecules. So automatically that will be suitable. This drug is suitable for the I mean actual meaning is this drug is interacted with these 3 protein molecules effectively, these 3 genes effectively. So automatically corresponding upregulations can be controlled properly by using that drug. I think you have got some idea about, right, by making use of molecular networks, how to proceed the drug designing. So you will be able to get some clear-cut idea about which all targets are to be used. Then that means screening of targets, identification as well as screening of targets properly. Then if gene is going to be used, again you can ensure that that target is very suitable for your work. So you will be able to do the interactome study. I think that interactome study it might be completed by this time. Now it is going to be over. 16th work is going on. So thereafter you will see that it generates all these networks completely. So protein network. Then actually you know that intact is actually an interaction network. So it's a very holistic, complete network will be there.